Hi, my name is Abigail,
Hi, I'm Jessica Smith.
and we're going to be talking today about developmental genetics and immunogenetics and chapter 22 in your book. So, our classic paper focused on mutations affecting segment numbers and polarity in the fruit flies. The study of the metametric nature of the patterns in the thoracic and abdomen regions of the larva epidermis of fruit flies. Very little is known about this for any organism about the genes involved in the process of spatial organization. To fully understand this concept, we need to know what it is and what it means. It's observed when components of an abiotic or biological group are arranged non randomly in space or on a larger scale, not related to genes or genetics, but think of ripple formations in sand dunes or patterns of waves. So just continue on to the found paper. Two basic mutations are described in this paper. The maternal effect mutations, where the embryos develop two posterior ends arranged in mirror image symmetry and lack a head, thorax, and anterior abdomen. This subdivision of embryo into segments is thought to occur by a differential response of the zygote genome to the maternal gradient. Homeotic mutations change the identity of individual segments. The ultrabiothorax transforms into the metathoracic and abdominal sections into the mesothorax segments. Pattern formations in the fruit fly. Each gene serves as a different function in the stages of early development in fruit flies. The establishment of the main body axis is the egg polarity gene. Determination of numbers and polarity of body segments is the segmentation gene. The establishment of identity of each segment is the homeotic gene. So moving on to the discussion, homeogenetic genes determine the identity of individual segments of, in this case, the fruit fly. Mutations in this gene causes bizarre rearrangements of body parts. The classic paper looked at several different types of these mutations, which are kind of listed here on the side in this photo. Each one is different, as you can see. You've got gooseberry, even skipped, odd skipped, crupple, patch, paired, runt, and curbs, I think is how you say it. This is just a table pulled from our book, page 670, just segmentation genes in the fruit fly. Each one of these genes control the differentiation of embryo into an individual. The three classes act sequentially, so that means that they happen in order. So gap genes, parallel genes, segment polarity genes, each one of them have examples on the side of what they represent. So moving on to the book in chapter 22, development takes place through cell determination. Every multicellular organism begins life as a unicellular fertilized egg. This single-celled zygote undergoes repeated cell division, eventually producing millions or trillions of cells that ultimately create a complete adult organism. Each cell in an early embryo stage is totoponent, which means that it has the potential to develop into any cell type. This leads us to the process of cell determination. There were several hypotheses regarding how this process actually works and how the genetic information might be expressed, which resulted in many experiments with cloning plants and animals. Dolly the sheep, I assume we probably all know about Dolly. She was not the first sheep to be successfully cloned. She was, however, the first sheep to be successfully cloned from an adult cell, which no one thought was possible at the time. She was important to the research surrounding how cells change during development and whether a specialized cell, such as a skin cell or brain cell, could be used to make a whole new animal. I thought the photo was interesting. If you see on the side there, that's her surrogate mother. Her white face was one of the first signs that she actually was a clone. If she had been genetically related to her mother, she would have had a black face.
Okay, so the paper we selected is called The Immunogenetics of Disease Resistance in a Fish, a Comparative Approach. This study was used to prove the immunogenic mechanisms that fish carry and is anticipated to have an underlying disease-resistant trait. The research included in this study is to show the immunogenic response involved in disease or immune health traits and how they can be correlated within genetic differences such as survival challenges and immune responsiveness. So genes in an organism can be influenced by the environment. Oopsies. Oops. Survival challenges, including the external world where the organism is located or developed, and also the organism's internal world, influences factors such as hormones and metabolism. This comes into play with disease resistance because if the organism is in a highly populated population, it will be more prone to diseases being spread and eventually the disease entering the body. So in the experiment portion of the paper included two approaches to study the genetic influences of genetic resistance. One being selective breeding for changes in immune responsiveness, and another one being a pattern of resistance. Selective breeding was used especially when disease resistance is expected to be under polygenetic control. It focuses on families that were homozygous and only for those genes determining the immune response under selection. Pattern of resistance aimed at a full genetic analysis of host resistance and examined a large number of models, for example, mouse families. In this experiment, inbred lines of species were highly dependent on for success, and inbred lines are basically established by successful mating of related species for at least 20 to 30 generations. Classical crosses, including F1 hybrids, F2 hybrids, and back crosses, were used to demonstrate Mendelian inheritance. Following the classical crosses, linkage markers were used to map the Mendelian locus on the chromosomes. The methods used to control infectious diseases included hygiene, vaccination, medication, and eradication of infected populations. Also, it has been established that both cold and warm-blooded vertebrates' resistance to infection can vary by a wide range of pathogens. Genetics can also play a factor for disease resistance, but the host influences the outcome of the immunity. The immune response is regulated by major histocompatibility complex molecules at the surface of antigen-presenting cells, the T cell receptor, and surface immunoglobulin molecules on B cells. So this Venn diagram shows the overlaps of ups and down regulated genes among each fish with bacterial challenges. The number indicated up, which is red, and down, the blue arrow, shows the regulated genes in each category. You can see in the diagram how different bacterial challenges affect different types of fish, and all this goes into account on the different development genetics and disease resistance traits that these fish have. The BOZ-ABH mouse model exhibits a reproducing, relapsing disease that accumulates permanent neurological defects. The BOZ model was used for multiple sclerosis and is proved to show how an autoimmune tolerance eliminates relapse, but fails to stop progression in a model of MS. This is important because this model was mentioned in our paper as the best known example of antibody-mediated immunity by selective breeding. And basically what antibody-mediated immunity means is the involvement of the activation of B cells and secretion of antibodies when presented with a pathogen. When it is exposed to the chemicals released by the activator helper T cells, the B cell divides, which then produce daughter cells that differentiate into memory B cells and plasma cells. So our selected paper conclusions. The inbred lines of mice and also chicken have proved to identify the response to cellular pathogens when infections come into the body. The immune response for these models have been diverse, caused by different genetic factors. The genetic factors already mentioned, including population size and genetic transferring, were already mentioned in the paper earlier on. This paper relates the value of immune parameters and importance of family selection in the disease resistance traits in fish. And here are our references.